The human brain is a universe of its own, a labyrinth of billions of neurons connected in an intricate web. These neurons, the fundamental units of our nervous system, communicate with each other through a fascinating electrochemical language. This language is essential to our being, allowing us to perceive the world, think, feel, and act. At the heart of this neural communication lies a phenomenon known as the action potential, a rapid electrical signal that travels down the neuron's axon. This signal is not a flow of electrons like electricity in a wire, but rather a wave of depolarization and repolarization of the neuron's membrane, driven by the movement of ions. This intricate dance of ions is orchestrated by a remarkable molecular machine embedded within the neuron's membrane, the sodium-potassium pump. This pump tirelessly maintains an electrochemical gradient across the membrane, a delicate balance of sodium and potassium ions that is crucial for the generation and propagation of action potentials. In essence, the action potential and the sodium-potassium pump are the fundamental building blocks of our nervous system, the intricate mechanisms that allow us to experience the world around us and interact with it in meaningful ways. Imagine a single nerve cell, a neuron, stretching out its axon like a long, thin wire. This wire carries messages, not in the form of electrons, but as a special kind of electrical impulse, the action potential. It's a brief but dramatic change in the electrical charge across the neuron's membrane, a sudden depolarization followed by a rapid repolarization. In its resting state, the neuron maintains a negative charge inside relative to the outside, a state known as resting membrane potential. This difference in charge is maintained by the sodium-potassium pump, which constantly works to keep a higher concentration of potassium ions inside the cell and a higher concentration of sodium ions outside. When a neuron receives a sufficiently strong stimulus, it triggers a cascade of events that leads to the generation of an action potential. This stimulus could be a sensory input, like the touch of a feather or the smell of coffee, or it could be a signal from another neuron. If the stimulus is strong enough to depolarize the membrane potential to a certain threshold, voltage-gated sodium channels embedded in the membrane open up. These channels are like gates that are specifically sensitive to changes in voltage. Once opened, they allow sodium ions, which are positively charged, to rush into the cell. The influx of sodium ions disrupts the negative resting potential, making the inside of the neuron more positive. This depolarization spreads down the axon like a wave traveling along a rope. This is the action potential in motion, the signal that neurons use to communicate. Crucially, the action potential is an all-or-nothing event. If the stimulus is not strong enough to reach the threshold, no action potential is generated. But once the threshold is reached, the action potential fires with the same strength and speed regardless of the intensity of the stimulus. This ensures that the signal is transmitted reliably over long distances without losing its strength. Think of it like sending a digital signal, where the information is encoded in the presence or absence of a pulse, rather than its amplitude. The sodium-potassium pump plays a critical role in this process by restoring the balance of ions after an action potential. It pumps sodium ions back out of the cell and potassium ions back in against their concentration gradients, ensuring that the neuron is ready to fire another action potential when needed. The electrical potential difference across the neuron's membrane, the resting membrane potential, is not just a passive state. It's an actively maintained condition, a delicate balance of ions and charges that is crucial for the neuron's ability to communicate. This polarization is maintained by the sodium-potassium pump, which acts like a tiny, tireless machine embedded within the cell membrane. It constantly pumps 3 sodium ions out of the cell for every 2 potassium ions it pumps in. This seemingly unequal exchange is essential for creating the electrochemical gradient that drives the action potential. The pump's continuous action ensures that a higher concentration of sodium ions is maintained outside the cell and a higher concentration of potassium ions inside. This difference in ion concentration creates both a chemical gradient and an electrical gradient across the membrane. The chemical gradient arises from the tendency of ions to move from an area of high concentration to an area of low concentration. The electrical gradient, on the other hand, arises from the attraction of opposite charges and the repulsion of like charges. The sodium-potassium pump harnesses cellular energy in the form of ATP to move these ions against their concentration gradients. This active transport mechanism is essential for counteracting the passive diffusion of ions through leak channels in the membrane, which would otherwise dissipate the electrochemical gradient. The maintenance of this gradient is crucial for several reasons. Firstly, it establishes the resting membrane potential, the baseline electrical state of the neuron that allows it to be excitable. Without this gradient, neurons would be unable to generate action potentials. Secondly, the electrochemical gradient created by the pump provides the driving force for the movement of ions during an action potential. When sodium channels open, sodium ions rush into the cell, driven by both the concentration gradient and the electrical gradient. This influx of positive charge depolarizes the membrane, leading to the rising phase of the action potential. Similarly, the efflux of potassium ions during the falling phase of the action potential is also driven by the electrochemical gradient, in this case, potassium ions moving out of the cell. The action potential is not a static event but rather a dynamic process, a cascade of ionic movements and voltage changes that propagate down the axon. This propagation is often compared to the way a flame travels along a fuse, with each segment igniting the next. As sodium ions rush into the cell during depolarization, they spread laterally within the axon, depolarizing adjacent segments of the membrane. This depolarization, in turn, triggers the opening of voltage-gated sodium channels in the neighboring segments, further propagating the action potential. This process continues down the length of the axon, like a chain reaction, ensuring that the signal travels in one direction, from the cell body towards the axon terminal. The speed of this propagation can vary depending on the diameter of the axon and the presence of myelin, a fatty substance that insulates the axon and speeds up conduction. The action potential is an all-or-nothing event, meaning that it either fires with full strength or doesn't fire at all. This is because once the membrane potential reaches the threshold, the positive feedback loop of sodium channel opening and depolarization ensures that the action potential reaches its peak. After the peak of the action potential, the neuron rapidly repolarizes, returning to its resting membrane potential. This repolarization is driven by two main factors: the inactivation of voltage-gated sodium channels and the opening of voltage-gated potassium channels. The inactivation of sodium channels prevents further influx of sodium ions, while the opening of potassium channels allows potassium ions to flow out of the cell, driven by both the concentration gradient and the now positive electrical gradient inside the cell. This outward flow of positive charge repolarizes the membrane, restoring the negative resting potential. During repolarization, the neuron enters a brief period called the refractory period, during which it is difficult or impossible to generate another action potential. This is because the sodium channels are inactivated and need time to recover before they can open again. The refractory period ensures that the action potential travels in one direction down the axon and prevents the signal from reverberating back on itself. It also limits the maximum frequency of action potentials that a neuron can fire, preventing overexcitation. Throughout this entire process, the sodium-potassium pump works tirelessly in the background, diligently restoring the ionic balance that was disrupted by the action potential. It's like a dedicated stagehand resetting the stage after a dramatic performance, ensuring everything is ready for the next act. This pump is not just a passive bystander, it's an active participant in neural communication, ensuring the neuron is ready to fire another action potential when needed. Without this constant maintenance, the electrochemical gradient would dissipate and the neuron would lose its ability to communicate. The sodium-potassium pump's role extends beyond just restoring ionic balance. It also contributes to the regulation of cell volume, maintaining the osmotic balance of the neuron, and plays a role in other cellular processes, such as nutrient uptake. Its significance is highlighted by the fact that it consumes a significant portion of the brain's energy budget. It's estimated that the sodium-potassium pump accounts for up to 20 to 40% of the brain's total energy expenditure, a testament to its crucial role in neural function. The energy expended by the sodium-potassium pump is not wasted. It's an investment in the brain's ability to process information, control movement, and generate thoughts and emotions. This remarkable molecular machine is a testament to the elegance and efficiency of biological systems. The action potential and the sodium-potassium pump are not just abstract concepts in neuroscience, they are the fundamental mechanisms that underlie our every thought, feeling, and action. They are the sparks of life that animate our nervous system, allowing us to experience the world in all its richness and complexity. Disruptions in these processes can have profound consequences, leading to a wide range of neurological and psychiatric disorders. Understanding how these processes work is crucial for developing treatments for these conditions and for unraveling the mysteries of the human brain. The study of the action potential and the sodium-potassium pump is a journey into the heart of our own being, a quest to understand the language of our nerves and the symphony of electrochemical signals that make us who we are. From the simple act of feeling a pinprick to the complex process of composing a symphony, every aspect of our existence relies on the intricate dance of ions across neuronal membranes. The action potential, that fleeting spark of electrochemical energy, is the fundamental unit of this language, the code by which our neurons communicate. The sodium-potassium pump, diligently working in the background, ensures that this code can be transmitted faithfully, maintaining the delicate balance of ions that allows for the generation and propagation of action potentials. It is a tireless guardian of our inner world, ensuring the smooth flow of information throughout our nervous system. As we delve deeper into the complexities of the brain, we begin to appreciate the elegance and sophistication of these mechanisms. The action potential and the sodium-potassium pump, once merely theoretical constructs, come alive as essential components of our being, the very essence of our thoughts, emotions, and actions. The study of these processes is not just an academic pursuit, it is a journey of self-discovery, a quest to understand the fundamental principles that govern our existence. By unlocking the secrets of the action potential and the sodium-potassium pump, we gain a deeper appreciation for the miracle of the human brain and the intricate tapestry of life itself.